Hi, I'm Dr. Jayesh Rao, Assistant Professor, Department of Botany, SKSV College, Lanza. Today we are going to discuss about polymerase chain reaction, that is PCR. Instead of using te- cloning technique to generate enough DNA for analysis or any other purpose, PCR is the alternate way to get more number of copies of desired DNA or gene. We have seen various DNA cloning libraries. DNA cloning takes weeks or even months to generate more copy numbers of DNA. We can use temperature cycling and enzymatic replication to generate millions of copies of DNA in a fraction of time. This is the idea behind PCR. Now we will discuss about history of polymerase chain reaction. The idea of polymerase chain reaction came up in 1983 when Karimulis, a scientist working for a CITES corporation in Northern California. The year was 1983. Biochemist Karimulis was driving one night when he struck with a flash of inspiration, stopping in a row. He wrote the idea of PCR on a back of receipt. He introduced PCR into the scientific community in 1985 at a conference in October, where CITES also rewarded Karimulis with $10,000 bonus for his invention. PCR technique, which is also called a DNA photocopier, is an in vitro technique that uses a few basic everyday molecular biology reagents to make large numbers of copies of a specific DNA fragment or specific region of a DNA strand in a test tube. The process is carried out in a PCR machine, requires DNA template, primers, tag or other polymerases, deoxynucleosides, triphosphates, buffer solution and divalent cations, for example magnesium, to run PCR, to run reaction. Now the machine of PCR is based on one principle, that is Peltier effect. Peltier effect is a heat exchange that results when electricity is passed across a junction of two conductors. Conduction electrons have different energies in different materials, and so when they are forced to move from one conductor to another, they either gain or lose energy. This difference is either released as a heat and absorbed from the surrounding. What is the overview of PCR? It is enzymatic DNA amplification, need to short sequences on the DNA, repetition of 30 to 35 cycles of 3 strips. A general overlay of PCR protocol: extraction of DNA, selection of target gene, primer designing for target gene or DNA fragments, then setup of PCR, amplification of target gene or DNA using PCR, visualization of results on gel or screen or sequencing, and the interpretation of results. Now what you need for PCR? First is sample or target DNA template, then primers for the specific detection, a single primer or pair of primers, nucleotides, that is mixture of DNTPs. Reaction buffer contains salts and other components to maintain ionic concentration. Magnesium included sometimes, enzymes, Taq polymerase or other types of enzymes like PFU polymerase. Now components of the PCR mixtures: deoxynucleotide triphosphates, deoxynucleosides, triphosphates containing phosphate groups, the building blocks from which the DNA polymerase synthesizes a new DNA strand. Equal amounts of each of the 4 DNTP, that is DATP, DCTP, DGTP, DTTP, are included in the reaction, usually at a concentration of 0.2 millimolar, that is 200 micromolar each. Now thermostable polymerase enzyme, Taq polymerase, Taq polymerase or another DNA polymerase with a temperature optimum at around 70 degrees centigrade, initially purified from the Thermus aquaticus and used in its native form. It was soon replaced by the recombinant version made from the cloned thermostable aquaticus gene expressed in E. coli. Now, primers. Those primer either use it in single or in pair. Now, buffers to maintain pH. Buffer solution providing a suitable chemical environment for optimum activity and stability of the DNA polymerase. Most buffer contains tris at 10 millimolar and KCl at 50 millimolar concentration. The pH varies from 8.2 to 9 at 25 degrees centigrade. So pH 9 at 25 degrees centigrade translates to approximately pH 7.6 at 72 degrees centigrade, which is the optimal temperature for polymerase activity. pH 7 to 7.5 is considered optimal for Taq polymerase. Now, magnesium chloride. Magnesium is a cation that binds to the polymerase and is an essential cofactor for polymerase activity. The lower the magnesium concentration, the more the stringent the conditions for primer annealing. A change of 0.25 millimolar can mean the difference between excellent yield of product of the expected length or no product at all. Now, stabilizers. In order to stabilize the polymerase, bovine serum albumin, that is BSA, 0.01%, gelatin 0.01% or glycerol 1 to 2% are sometimes included in the reaction mixture. Then enhancers. A number of chemicals can be used to optimize the performance and sensitivity of PCR, including dimethyl sulfoxides, DMSO, that is tetramethyl ammonium chloride, that is TMAC, and betaine. In effect, this lowers the temperature of the annealing step. DNA template. However, the cleaner the template, higher the sensitivity. The monovalent cations. Monovalent cations, potassium ions, a standard PCR buffer contains 550 millimolar KCl and work well for the amplification of segment of DNA 500 base pair in length. Divalent cations. All the thermostable DNA polymerase requires free divalent cations, usually magnesium 2+ ions, for activity. Calcium ions are quite ineffective. Magnesium or manganese ions, generally magnesium 2+ is used, but magnesium 2+ can be utilized. Manganese 2+ can be utilized for PCR mediated DNA mutagenesis. As higher manganese 2+ concentration increases the error rate during DNA synthesis. Although a concentration of 1.5 millimolar magnesium is routinely used. Inhibitors. Almost anything will inhibit PCRs if present in excess. This includes proteinase K, phenol, and EDTA. Other substances that can cause problems are ionic detergents, heparin, polyanions such as spermidine, hemoglobin, and gel loading dyes such as bromophenol blue and xylene cyanol. Now, critical parameters in PCR, that is annealing temperature of primer. The ideal annealing temperature of particular primer is related to its length and sequence. That is the content of or amount of G+, guanosine and cytosine bases. If the temperature is too low or magnesium concentration is too high, then the primers can bind to non-specifically to the DNA template. At annealing temperature, the primers are in movement caused by Brownian motion. Ionic bonds are constantly formed and broken between a single stranded primers and template. When primers comes in contact with a perfectly complementary target sequence, the bond that forms is sufficiently stable to allow DNA polymerase to initiate DNA synthesis. Now, steps of PCR. apparatus to perform about 35 cycles of a 3 temperature procedures. First is 95 degrees centigrade step. Whole is a initial denaturation of DNA. Whole genome will convert in a single stranded. The next step is 95 to 94 degrees temperature denaturation, 50 to 60 degrees centigrade annealing of primers, binding of primers to template. Then next step is at 72 degrees centigrade, that is elongation of the primers, that is elongation of chain formation, and 72 degrees at same temperature, that is 72 degrees centigrade extension. The process of PCR. The first is separating the target DNA, that is denaturation. The tube containing the sample DNA is heated to more than 90 degrees centigrade Celsius, which separates the double-stranded DNA into two separate strands. The second step is binding primers to the DNA sequences. This step is called annealing. One or two primers are used in step 2. The primers bind to the beginning of the sequence that will be copied, marking of the sequence for step 3. That is, during this steps 2, the tube is cooled and primers binding occurs between 40 and 60 degrees centigrade. Now making a copy. Third step is making a copy, that is extension. The temperature is increased to approximately 72 degrees centigrade. Beginning at the regions marked by the primers, nucleotides in the solutions are added to the annealed primers by the DNA polymerase. After completing the extension, 2 identical copies of the original DNA have been made. Now the final elongation. This single step is occasionally performed at a temperature of 70 to 74 degrees centigrade for 5 to 15 minutes after the last PCR cycle to ensure that any remaining single strand DNA is fully extended. This is logarithmic multiplication. This is PCR through edges. Thank you.